Hi, I'm Meredith. I'm a dietitian. I might not be your dietitian, so please talk with your own personal health care provider before you make any changes to your diet, your supplements, or your medications. This video is to inform, and it's a video that I actually started. It's 3 videos. I started with this about 3 weeks ago when I was trying to explore whether or not anhydroretinol was the destructive force behind my own family's health issues as well as my clients. And so I wanted to map out what happens in the body when we have too much anhydroretinol. And so this is the first video, and it's going to talk about vitamin A metabolism and sources of apocarotenoids and anhydroretinol. It's a bit of a review, but also I would make sure to, I kept reading information. And so I wanted to make sure to give the most updated information. So that's why I decided to add this to this video series, just to give updated information. Okay, so I'm going to go over the metabolism of vitamin A just a little, so that we have a good understanding of how it's metabolized in the body. So we eat vitamin A in the form of retinol esters, and this is the animal source of vitamin A. So it'd be in things like butter and cheese and milk. And it's also an animal meat, although I've heard that that's a big controversy, that I posted a video about animal meat having more retinol than we actually think. So that's kind of fun. Yeah, so sorry for rocking the boat. So we can actually consume this, and then in our intestinal tract, we break it down. We take a fat off of it so that we can absorb the retinol, which is a small polar molecule. We can absorb it into our cells through the cell membrane, and then we repackage it into chylomicrons. The chylomicrons, they enter our lymphatic system. They don't go straight to the liver. They enter our lymphatic system. That empties in next to our heart, and then our heart pumps and it pushes all of the chylomicrons through the whole body. And this happens about 3 to 4 hours after a meal. And then we have hormones, lipoprotein lipase. It takes the triglycerides off of the chylomicron, and then you can access the retinol. And then whatever retinol is left in the chylomicron will go to the liver, and the liver will store it in hepatic cells or use it as needed. So now we know that, thanks to that, to a quantum mechanics paper about the role of vitamin A in the body, we know that retinol plays a huge role in mitochondrial health. And so the liver will use retinol for that, and then whatever is not needed will be metabolized away. So, we have, whenever we need to access retinol, we have retinol ester hydrolase, and so that is what removes the fat. Whenever we need to package it back up to store it in the liver, we have lecithin retinol acetyltransferase, and this is choline dependent. So you need a source of choline. If you don't eat choline, then you'll take it from your cell membranes, and that's something important to remember. We don't want to break down our cell membranes to be able to store vitamin A safely. Yes, you can exceed the capacity of your liver to store vitamin A. So safely is the key word there. So if you need to metabolize retinol to another form that's used in the body, there's enzymes that do this, and they're NAD dependent. But also some of, you can use NADPH too. But basically, you require a cofactor to be able to go from retinol to retinol aldehyde. And retinol aldehyde is the form that we typically see, or we know we use in our eyes to perform the visual cascade. But I believe that too much retinol aldehyde, like if you get stuck here, that can cause a thing called a lipofuscin. And lipofuscins are a combination of ethanolamine and retinol aldehyde, and makes A2E. And I think that this is a contributor to dementia. A2E aggravates microglia cells, so I think that it causes damage in the brain, basically. So Also, A2E can cause mitochondrial damage as well. And so I don't think that all the forms of vitamin A are off the plate. I think if you accumulate in any particular form, you have an issue. So it's all about metabolism. Okay. So when we go from retinol aldehyde to retinoic acid, that's also NAD dependent. And it's by, we use enzymes ALDH1A1 or retinol aldehyde dehydrogenase. And our backup when we don't have enough NAD is AOX, and it's molybdenum dependent. That's supposed to be an O. I keep forgetting to change it on my slides. So, and I might be saying molybdenum wrong. I think everyone knows I have a problem with that word. I have like mind block against it. So retinoic acid is metabolized to smaller versions of it so that it can be excreted. And the enzyme that does that is CYP26, and it's NADPH dependent as well. As well. Okay. All right. So now let's move on to, one more thing about this. If you are not able to make good CYP450 enzymes, so there's people who have problems making heme, and there's lots of conditions that can cause that. If you're really bad at making enzymes, or potentially you have a slow CYP450 enzyme activity, retinoic acid can build up, and then retinoic acid will stop. It'll have a feedback mechanism on ALDH, and it will stop you from being able to metabolize retinol aldehyde, because retinoic acid in a large amount is poison and it will hurt you. And so your body's very smart about inhibiting its own production. So in Accutane poisoning, that's retinoic acid. So that's something, it could be a trigger for problems with vitamin A metabolism, because you would end up starting to accumulate more and more of the precursors for retinoic acid. And that, I think, is a contributor to this issue that I'm talking about with anhydroretinol. Okay, so beta carotene is our vegetable source. Beta carotene needs to be metabolized either to retinol aldehyde or to apocarotenoids. We don't want to have straight beta carotene in our bodies because it can be toxic to mitochondria. So the first enzyme, BCO1, it's in the intestinal tract, and some people have very slow BCO1, and that means that they're not good at making retinol aldehyde or retinol from beta carotene, because you got to get to retinol aldehyde first. So people with slow BCO1, they're not very good vegans. Like, there's people who become vegans and they are amazing. I was a vegan for like 30 years when I was an environmental science major, and I was the sickest vegan. I was horribly sick. So then there's the, people who are vegan and they struggle with obesity. I think that those people are probably have a slow BCO1 also. And so, if you don't have a healthy amount of retinol, now we know that you have to have it for your mitochondria to work correctly in oxidative phosphorylation. So that kind of explains the obese phenotype of veganism. Also, iron deficiency can play a role in that. So there's another thing. Lots of things that could go into that. Okay, so so you have beta carotene. And then if you have a slow BCO1, you absorb more beta carotene, but you have this backup sort of enzyme called BCO2 that's in the mitochondria, and its goal is to keep you from having toxic levels of beta carotene. So it also metabolizes beta carotene, and one of the metabolites is beta apo10 carotenoid. And this one is a metabolite that was in the quantum mechanics study on retinol or retinoids, and it has been shown to act like anhydroretinol in the PKCδ signalome. So that's important to know. So if you have a slow BCO1, you have a higher chance of making this particular version of an apocarotenoid. And that's not necessarily bad if you have enough retinol around. It could actually be helpful. They say in the study that it could be beneficial to the heart. It could give your heart an energy boost if you have enough retinol to buffer it. Okay, so then also we can make this other form of beta-apo13 or beta-apocarotinol, or it's actually carotinone this time. So this particular one, I've not seen anything that it can act like anhydroretinol. I haven't seen that, but what it does do is this is made under oxidative stress. So I believe that people who are making anhydroretinol, which anhydroretinol causes a pathological activation of pyruvate dehydrogenase complex. I believe people who are making this end up making beta-apo13 carotinone because they make reactive oxygen species in a large amount. And so you can make this from all of the all retinol, aldehyde, retinoic acid, and retinol can make beta-apo13 carotinone. And the problem with that is that this actually inhibits CYP26. So if that particular, metabolite of retinoids inhibits CYP26, then you have a buildup of retinoic acid, and retinoic acid will shut down your ability to metabolize vitamin A, and that can lead to an accumulation of retinol or an accumulation of retinol aldehyde. And if the A2E complex can mess with your mitochondrial respiratory chain, it can cause damage. So yeah, that's not a good situation, right? So we want to get out of a high reactive oxygen species situation. We want to get better at vitamin A metabolism. I do not believe that all vitamin A is a poison. I do believe it's dose dependent. I do believe that a lot of the dysfunction is from anhydroretinol. I would love to do studies on this. So, all right. So sources of anhydroretinol. You could get this from your diet. It's variable depending on how the vitamin A supplements are made, or because they're adding these to products. So vitamin A acetate, I believe, would be the worst type of supplement because acetate is in solution, dissociates to acetic acid, and acetate's like a little dissociation constant, and acetic acid is a catalyst for the conversion of retinol to anhydroretinol in the presence of alcohol. So we do make alcohol in our body. We make about 4 grams a day in digestion. So we also make it in our cells during metabolism. So yeah, I don't know if this is necessarily a good thing. So check your supplements. You want to look for something that has, I don't think you should take a vitamin A supplement. Let me say that. Unless your diet is completely void of vitamin A, then maybe you should, or maybe you're tracking it, and maybe you have like anorexia and you haven't eaten in years. It's possible. It's because you do need vitamin A to run your immune system. Like your gut associated lymphoid tissue, you have to have some vitamin A in there to be able to make IgA, which is an immunoglobulin that helps you to fight off pathogenic bacteria in your gut. So we do need some vitamin A. Okay. So you want to watch out for vitamin A fortified dairy products. Some of them might contain anhydroretinol. Never buy a milk that's in a glass bottle that's clear, because ultraviolet light itself can have an effect on retinol. Enteral formulas and oral nutrition beverages, and even baby formula. We need to be careful about the exposure of it to light, because all you need is a little bit of alcohol. And I believe so over time in formulas, studies show that the vitamin A degrades over time. And what that is that the palmitate, the fat, comes off of the vitamin A. And so the retinol is just kind of free in solution. And so retinol itself is a primary alcohol. So if there is a primary alcohol around and an acid, then there's an issue. You could make anhydroretinol. And that's not good. So one safe mechanism or safety thing to do would be to have all of your products that have vitamin A in them, have them in containers that are not clear, because light is also involved in this process. And these are just ideas that I've had about how this is happening. Of course, it would have to be studied in a lab. So, all right, so shelf-stable products would also potentially have some anhydroretinol, because inherently when you make a vitamin A supplement, there is some of this in it. Sometimes some of the studies show like 3%, but 3% might be okay for one person, but not for another if they have broken metabolism. And skincare products that contain retinol, when you put them on and you expose it to light, you start to have a problem. You make that anhydroretinol. So I think the biggest sources of this, though, are formation in the gut before absorption because of dysbiosis. If you have aspergillus or candida or other, fungal issues, fungus like to make alcohol, and alcohol plus an acid such as hydrochloric acid or acetic acid, then that at a human body temperature makes anhydroretinol. And we know also that we make it during metabolism because they've identified that this is made in lymphocytes. And they also use anhydroretinol for as to kill cancer. So it's not like a small amount is bad. I think what's happening is that there's an imbalance and there's way too much anhydroretinol. Cod liver oil was the first place that they found anhydroretinol. So I don't think I would take cod liver oil. And the majority of people that not majority of people, but large quantity of people in the vitamin A toxicity group on Facebook, they did have cod liver oil, and it was a contributor to getting stuck in this pathway. And then freeze-dried liver, I think would be not a good idea because it's just sitting over time. Again, there's like a degradation issue. And frozen liver, I'm not so sure. I think fresh would be the best way, although liver, you don't need very much. The RDA for vitamin A, like for your whole day, and RDA meets 98% of the population's needs. So it meets the people's needs who have low needs all the way up to high needs. It meets the person with the highest needs. You only need 1/4 of an ounce for an adult. That's like nothing. So I would be very cautious about liver as a source of vitamin A, unless it's the only thing you tolerate. So on a frozen liver, I mean, if you think about it, if you reduce the temperature, then less anhydroretinol would form. But how much anhydroretinol was in that liver to begin with? So same thing with regular liver, right? Like how much is in there? We don't have the testing available to figure this out. So, okay, so skincare products. So if you have a skincare product that has vitamin A added to it, within 15 minutes of being an ultraviolet light, visible light, you can end up, and that includes like our LED lights. It's not just outside. So like I have like this wake up light over here, just exposing my skin that has retinol on it, I'll make anhydroretinol. And so it would be in my skin. So that's not good, right? Okay. So what about in the body? I think the major issue here would be that is the state of the alcohol in the body. Okay, so alcohol, you have to have alcohol to make anhydroretinol. So it's just an acid alone wouldn't work. It wouldn't be able to dehydrate to remove this hydroxyl group from retinol. You have to have an acid. So in laboratory experiments, you can actually make anhydroretinol at 68 degrees Fahrenheit with ethanol and hydrochloric acid. But there's studies showing too that you can use ethanol and acetic acid, which is so hydrochloric acid, pKa is -6. It's a very strong acid. But acetic acid isn't so strong. pKa is 4.8. But the temperature that you need to be able to make acetic acid make anhydroretinol is 98.6 degrees Fahrenheit, which is human body temperature. Pretty weird, right? So other acids that are potential at 98.6 degrees Fahrenheit might be oxalic acid. So selenic acid. So selenic acid, sodium selenate is added to supplements as a form of selenium, and that's selenic acid. So that's something to look for on your supplements. You want to look for any of the things that say 8 on them, because 8 is actually the acid when it dissociates in water. I don't want to scare you about everything, but just be aware. For some people, every little thing counts, and for some people, it doesn't. That's how we notice that there's differences in tolerations of different types of supplements, right? So hydrochloric acid, obviously. So if you have candida in your stomach or your esophagus, then that is producing alcohol. And then you have your stomach acid, and an acid plus alcohol makes anhydroretinol. Sulfuric acid too, but so that would be sulfate. If sulfate was accumulating, then it's a very strong acid. You would only see this hopefully, and well, not hopefully, sadly, sadly in chronic kidney disease. You start to retain sulfates. So these are all the things that I think could be contributors. I think that the biggest one that most people are dealing with is probably a gut issue. Although, like I said, intracellular metabolism within the body is a big thing too. I think that's all I want to say there. Yeah. Okay. So how do we get out of that, right? We have all these forms of vitamin A, all trans-retinoic acid, retinol, anhydroretinol, 14-hydroxy-4, 14-retroretinol, 13-14-dihydroxyretinol, and then we have rehydroretinol. So how we get out of it is we control alcohol. That's what I'm thinking. I think that that's the number one issue, is that we need the absence of alcohol. Definitely lower amounts of acids would be helpful. Not hydrochloric acid, we need that for digestion, but like within our body, we want to not be making excessive amounts of acids that could produce anhydroretinol, like acetic acid. So if you don't have alcohol around, then your body will take anhydroretinol and rehydrate it to rehydroretinol. And when they found this they did this in studies, it was animals that were deficient in vitamin A who would make rehydroretinol. So vitamin A itself, retinol, is a primary alcohol. So that's something to consider, is perhaps very, very high vitamin A diet could be causing a very high level of unbound retinol. And that's something I'll explore in future videos. Okay, so if we make rehydroretinol, though, if you look at retinol, look at the double bond formations. And this is like the beta iodine ring, beta iodine, I think is how you say it. And the double bond on this, there's a double bond here. And then there's also a double bond right next to the beta iodine ring. But when you make rehydroretinol, the double bonds have moved. And so it's not an efficient form of retinol. I don't think that it probably works really well in metabolism. Probably not a good thing for the signalome. It's probably an inefficient way to monitor the energy levels in a cell, like in oxidative phosphorylation, that we'll talk about in the next video. So I think that our real big goal would not to be make rehydroretinol. It would be to prevent the formation of anhydroretinol. There was another study that showed that anhydroretinol can be converted to this 14-hydroxy-4 retroretinol. And anhydroretinol, 14-HRR it can help cells grow. I think this was lymphocytes. It can help lymphocytes stay alive, but anhydroretinol kills them. So that's kind of interesting, right? It's like it's good versus evil. And then we have meh. So evil anhydroretinol, good 14-hydroxy-4, 14-retroretinol, meh, rehydroretinol. It's just like the meh. Okay, I'm sorry. It is really early in the morning. So the next video is going to be about massive mitochondrial damage, and it's why I decided this is the issue. And so working through those pathways is the reason why I decided, hey, this is it. I'm just going to share this and be brave and take a leap of faith and be probably attacked by the wolves, because that's how it goes in science. And I'm okay with being attacked sometimes. Sometimes I'm not. Sometimes I'm very discouraged. So I'll see you in the next video.